Anyone who's joining us. Dr. Morse, can you hear me okay? Thumbs up if you can. Thumbs up? Okay, great. So sorry about that quick resistance with the feedback on the line. We're really excited this morning to have some open dialogue discussion around immunotherapy and colorectal cancer. And so we're going to go ahead and get started with our slide deck in just a second. But just a quick note that as we're moving into today, that this platform is a little different. We were just trying different options as related to having some discussions during webinar type programming. So in today's session, we are going to hear from Dr. Mike Morse, who's been a part of our immunotherapy team. And Dr. Morse is really going to talk about the signature and hallmark of work that's relevant to colorectal cancer and immunotherapy. And we're going to take time to also talk about Fight Colorectal Cancer's connection to the work and where we're going as a result. But I think we want to definitely spend time to hear from the critical mass, all of you, through the chat feature. If we can work it out to open up a mic to have people answer and ask questions, that's what we'll do as well. So just a quick note is on the bar on the side, much like the GoToMeeting, there is an opportunity down below to go ahead and put in the chat feature any thoughts, questions that you might have related to the webinar today. If you want to catch today's webinar, we'll go ahead and archive that in the Fight CRC website. And we are hosting this today at the University of Colorado School of Public Health. So special thanks to Ashley and to Reese and Kirsten, who are part of our combined School of Public Health and Fight CRC team members, as well as Michael Sola, Danielle Burgess, and Elizabeth, and Andrew, and many others who are helping support the great work for today as well. So on the right side, or actually I think we're below where you can hit the chat feature on the bottom of the deck. We're excited to move this forward. So just a quick note that a reminder that on the Fight CRC website, in addition to today's webinars, there's a cadre of mini magazines, the very popular Guide in the Fight, as well as the Tabuti podcast series that are recorded as well as great resources for you to utilize for prevention, detection, treatment, survivorship care, palliative hospice care as well. So a lot of great resources. So just as a reminder, and I think we've gone through this many a time, we want to reserve today for general information and education. This is no place, really a specific place that's quite a clinical design of sort. We really reserve the time in education to be general background, but not to ask specific medical questions. And if there is an emergency, we ask that you call your provider or 911 system. So with that, we're going to go ahead and start just a quick overview. As I mentioned today, we're going to hear from Dr. Mike Morse, who's going to give us really at the state of immunotherapy as it relates to colorectal cancer. And Mike was part of our advisory team and a key person who helped develop the manuscript that Fight CRC developed in concert with the Cancer Research Institute over the last year and a half. And so Dr. Morse was one of the driving force in the 2025 member convening group, which represented academic, bench science, industry, as well as nonprofit organizations. So we're really excited for Mike to share that perspective. After Dr. Morse is done presenting, I'm going to spend a bit of time talking about specifically the outcomes of our convening and what this paper held from a more practical standpoint, and then we're going to really take the remainder of the time to delve into questions. So again, it's going to be critical that you key in any questions that you have. If the group isn't too big, we can see about opening the mic, but I'm not sure just yet how that's going to work. And then we'll wrap up quickly. So Dr. Morse, I think, Ashley, he is unmuted. Dr. Morse, let's just make sure that we're set and ready to go. So Dr. Morse, we'll go ahead and advance slides for you today since we just received your most updated PowerPoint presentation, but I think you have the mic. So Mike, if you don't mind giving us a little bit of background, I mean, folks have the slide in front of you, but I think it's also great for you to introduce and give some background about who you are, in addition to, of course, your academic and science background here.
Sure, happy to, and thanks again for having me here to present this and to be part of the project. So I'm a medical oncologist. I specialize predominantly in GI oncology and also do see some melanoma patients. And I've been interested in immunotherapy for my entire career. Back when I was a fellow, we started working with models of cancer vaccines, and we're one of the first groups to perform dendritic cell vaccine studies. And we've continued to be interested in vaccines as well as combinations with immune modulators, and have carried those through phase 1, phase 2, and some phase 3 involvement as well. In particular, I've been very interested in colon cancer as a target because obviously it's a large number of patients that I see, and has continued to be a challenge for immunotherapy. But I think we're starting to certainly see some strides for the MSI patients for sure, but even for microsatellite stable patients. And we'll talk about some of that data going forward. Next slide. And I'm not sure if it's slow on my end here. Andi, okay, I'm sorry. Go ahead with next slide. The next two slides are going to show the cancer immunity cycle. This has been documented in many articles. It's a compendium of what the current thinking is about how the immune system responds to tumors, both being activated against them as well as responding and destroying them. So you can see at number 1 in the figure there that there's cancer cells are always releasing tumor antigen as they die. And those antigens are picked up by regional dendritic cells, which deliver the antigen to regional lymph nodes where T cells are located. And then those dendritic cells hopefully activate T cells that can recognize that tumor antigen. And then the T cells can spread throughout the body through the bloodstream, and they tend to home into tissues that are inflamed. And as they reach those tissues, if they can recognize tumor that has that same antigen being presented, they'll engage that tumor, and as long as they've been sufficiently activated, they should destroy the tumor cell, releasing more antigen, and a new cascade of T cell activation can occur. One of the challenges in this whole process is there's many different cell types. It's not just T cells, and there are many different cytokines and many different molecules all working together. Some of these cells and cytokines and molecules are activating. They help the immune response along and help T cells to destroy tumors. And some of these molecules are inhibitory. They lead to T cells that are not activated, or they lead to an inhibition of T cell interaction with tumors. And some of these molecules you can see on here are ones you've probably heard about. For example, CTLA4 is at the point when T cells are being activated, and it tends to cause T cells to downregulate their activation and their proliferation. And at the point where T cells are interacting with tumor cells, you see PD-1 and PD-L1, that interaction between the PD-L1 expressed by tumor cells and the PD-1 on T cells that causes T cells to undergo senescence and no longer be able to attack the tumor cells. But there are many other opportunities here. Next slide. That are places where we can interact with this cancer immunity cycle for actual therapeutic approaches. So at some of these same points, you can see that if you want to activate an immune response, you can use vaccines, you can use cytokines, activating antibodies. We don't have currently great ways of ensuring T cells will necessarily target to the tumors we want them to, but certainly once we have a T cell in the tumor environment, we have antibodies that prevent the tumor from engaging the T cell through the PD-L1, PD-1 interaction, so the T cell can attack. If we don't activate T cells that we're happy with, we can always supply those T cells through CAR T cell therapeutics. And there are many other molecules and targets in here that there are drugs in development for as well. Next slide. We're going to talk about colon cancer in particular and why we've had some success and why we hope for more success in the future for those colon cancers that haven't been sensitive to immune therapy. It's known for colon, as in most cancers, that there are T cells that can infiltrate the tumor, and the more T cells that infiltrate the tumor, in particular, not just the edge of it, but infiltrate into the tumor milieu itself, that there's a better outcome. So on the left-hand side, you can see that for primary colorectal cancers, if we find that there's a high T cell infiltrate, that the overall survival for those cancers is better. And this is being developed in a test called the Immunoscore, which we hope in not too distant future will be part of the staging of colon cancers. We won't just have TNM, we may have TNM and I. We look at the immune response, and that'll also help tell which patients are more likely to have a better outcome than those who don't. On the right-hand side is also a graph demonstrating that for metastatic disease, the same thing, that if you have a high T-cell infiltrate, in general, you have a better prognosis. You can see, though, that among the metastatic patients, that their colon cancers, although they do have T-cell infiltration, those that have very high levels of T-cell infiltration tend to be, let's say, a minority of those patients. Next slide. Now, among the colorectal cancers that are heavily T-cell infiltrated, we know that MSI high colorectal cancers are a unique subgroup. First of all, we've known for a long time that this concept of MSI high, and in the next slide I'll show you some more information about the molecular characteristics of it, but for right now we'll just call this a group that has a high mutational burden, that when the pathologist looks at them under the microscope, they can often identify them. They have high levels, very high levels of T-cell infiltration, what some people call a Crohn's-like reaction. Crohn's disease is an autoimmune disease that strikes the colon and small intestine, and often has a special pattern of lymphocyte infiltration. Also, these tumors are often appear to be poorly differentiated. They tend to be on the right side. Even though they're poorly differentiated, though, and you would think that would create a poor prognostic scenario, in fact, they have a better prognosis than other colon cancers. So you can see that in this slide that the stage of colon cancer also reflects the MSI contribution. Earlier stage disease tends to have more tumors that are MSI high, whereas once you get to metastatic disease, stage 4, not that many of them are MSI high. That sort of reflects the prognosis. Earlier stage tumors tend to have better prognosis. They're a little bit more often MSI high, and they tend not to make it to metastatic disease. But some metastatic disease will have MSI nonetheless. Next slide. So what's special about MSI high colon cancers, as you can see here, is they tend to have a high infiltrate of T cells. CD4 positive helper T cells, CD8 positive cytolytic T cells, the ones that actually do the killing. They also can have FOXP3 positive. Those are the regulatory T cells that tend to dampen the immune response. And you can see that this occurs both at the invasive front, where the cancer meets the immune response or meets the surrounding tissue, and also within the stroma of the tumor. We really want it to be in the stroma, if at all possible, because that's, of course, where a lot of the cancer cells are. But even the invasive front, there's some benefit to having T cells infiltrating. Next slide. Here's the molecular characteristics of MSI high, or some people call this DMMR, mismatch repair deficient. So the reason this occurs is that it's often that additional base pairs get inserted into DNA during replication of the DNA. The problem is, if you insert even one additional base into the DNA, it shifts all the other bases over such that the amino acid sequence is going to be different for that protein. Now, sometimes that leads to a protein that is truncated and has no function, but sometimes what that does is creates a new protein, essentially, that has a different amino acid sequence just by that insertion or sometimes deletion of an individual base. Therefore, it's really critical to have proteins that can fix that mistake so that you don't have lots of mutations in your proteins. And so this is done by a complex of 4 molecules, and you can see the names of them there: MLH1, MSH2, MSH6, and PMS2. They work together as a group to look for these mistakes along the DNA, these insertions and deletions, and to remove them and put in the proper base. If that doesn't happen, you get a very large number of mutations that occur. That is, if you're missing one of these proteins and you don't correct the mistakes, then anytime there's an insertion or deletion like this, you can have a mutated protein. And so this is manifest in several different ways. One, in the left lower hand, you can see that there are portions of the DNA, they're called microsatellites. We're not entirely sure what they do completely, but they're scattered throughout a person's DNA. They are very prone to these insertions and deletions of bases. And if you have a scenario where you have deficient mismatch repair, or the other terminology, MSI high, is that these microsatellites throughout the DNA tend to have different sizes than what they should have. They should all have the same length at a particular microsatellite. They should all have the same length if you have fidelity in DNA replication. But if you have insertions and deletions throughout the genome, these sites are very prone to that occurring, and they're very easy to identify. We've had the technology for a number of years to identify by PCR that at a particular microsatellite, there's sort of different lengths of the microsatellite instead of what should be a single length. And that's reflected here in what you can see is sort of a PCR gel that instead of having is just sort of a unique size, you have many different sizes. It can also be reflected if you actually sequence an individual gene. If that gene has these mutations in it, you can identify that it's completely mutated. There's one or more amino acids that are no longer what they were originally. And that is reflected in when those proteins get broken down and presented on the surface of the tumor cell in the MHC molecules. It's reflected in what are called neoantigens or neoepitopes. You no longer have the original amino acid sequence. Sometimes that mutated portion of that protein will be inserted in DNA and presented on the surface of the cell. Well, that is very immunogenic because T cells are looking for differences. They're not looking for the same DNA, the same proteins, They're looking for proteins that are different, that don't belong. And what you often find in MSI high cancers that have a very high mutational burden and many neoepitopes, you find T cells that can recognize these neoepitopes and can kill specifically those cancer cells. Next slide. I'm sorry, on my end, it's a little bit slower. So you would think that most MSI high cancers should have a very high T cell infiltrate. They do. You would think that they're very sensitive to T cell killing. They are. And that's the reason why the prognosis for MSI high cancers tends to be better, because the innate the adaptive immune response already present within that person is already recognizing and trying to attack the cancer. However, whenever you have T cells trying to attack a cancer, the cancers will try to protect themselves. And one way they do that is by deploying these checkpoint molecules, so, PD-L1 in particular. And LAG-3 is another one that's been seen. And if you look at either the invasive front or within the TILs and stroma, what you see in MSI high cancers are high levels of PD-L1 or T cells that express PD-1 or expression of LAG-3. All the checkpoint molecules, next slide, tend to be present within MSI high cancers. In other words, they're already trying to protect themselves from that very high level T cell response that's otherwise attacking. Still waiting for the next slide to load up here. Okay, so if you ask the question now, is there a way to intervene with those checkpoint molecules so that the T cells that are already present, that can already recognize the tumor, that should be able to kill, can now go ahead and kill these cancer cells? The natural question is, what if we use checkpoint blockade? And the slides here are from two studies. One is the sort of landmark study in this area by Young Lee, where patients with both MSI high, or so to speak, MMR deficient tumors, or MSS, or so-called MMR proficient tumors. Patients with these tumors were given pembrolizumab, the antibody that binds to PD-1 and prevents the PD-1-PD-L1 interaction. In the upper part of the slide on the left, you can see that the percentage of patients among those that had MMR deficient tumors who had either stabilization or shrinkage was quite high. That would be the green and the lower part of the graphic. If you now go to the graph at the lower left, you can see that the probability of improved progression-free survival was also better within those that were mismatch repair deficient, the MSI high patients. So not only do they have a response, that's a durable response, and therefore progression-free survival is quite a bit better than in those cancers that are MMR proficient or microsatellite stable. In a more recently reported study, which is still going on and reporting out some interesting data, there'll be even more data from this at ASCO GI, Dr. Overman's study with nivolumab. This was limited to patients that were MMR deficient, or again, MSI high. Again, you can see a very high rate of either tumor shrinkage or stability of disease in patients who had the checkpoint blockade. And again, this presents in a way that patients will have durable responses. So you can see the curves from this study are essentially plateauing now at a relatively short period of time, so that we know there are patients who are going to have very durable long-term remissions. Some are even complete responses, but you can see the disease control rate is very high at 70%. Okay, next slide. One of the questions that's come up is whether there are subgroups of patients who seem to benefit more in this group, and of those that are responding to checkpoint blockade. And the answer is we really haven't found that so far. PD-L1 expression does not appear to be critical. Those with low or higher expression in general can have benefit. If you look at those that have a high level of PD-L1 expressing tumor-associated immune cells, these are some of the immune infiltrating cells that are also thought to, if they deploy PD-L1, are thought to also participate in inhibition of T-cell mediated killing. If it's not the tumor PD-L1 expression, it's sometimes the infiltrating immune cells. That doesn't seem to be associated with outcome. You can respond either way. BRAF mutational status. Remember, many of the MSI high cancers have BRAF, which is often considered a poor prognostic factor. Doesn't seem to matter. You can still benefit. And whether you get to be mismatch repair deficient because you have Lynch syndrome, or whether it's sporadic, doesn't seem to matter. There's still benefit. Next slide. Well, this has led to the question, how do we take tumors that are microsatellite stable and make them like these MSI high cancers, at least in the high T-cell infiltration and in the sensitivity to checkpoint blockade? Because that's been the challenge so far. From Young Lee's paper, you saw that there was a low rate of response. In fact, there essentially are no responders to single-agent checkpoint blockade among microsatellite stable proficient MMR cancers. Well, one point is that many of the microsatellite stable colon cancers don't really have high levels of T-cell infiltration. And not only that, they sometimes have low levels of MHC or HLA expression on their surface. In other words, fewer molecules to even present neoepitopes if they exist within the tumor. So that led to some very interesting preclinical work, which was ultimately channeled into a preliminary clinical trial where it was observed that MEK inhibition with a drug called cobimetinib led to a higher level of T-cell infiltration within the tumor and higher levels of MHC expression on the cancers. In other words, MEK inhibition gave you a milieu that was very much like what you see in MSI high cancers, more T cells, more display of tumor antigen. And in preclinical models, if you combined a MEK inhibitor with an anti-PD-1 or anti-PD-L1 approach, you had the greatest efficacy, the greatest tumor control. Therefore, in a study reported by Johanna Bandel at ASCO, patients with microsatellite stable colon cancer and a high level of them having RAS mutations, a difficult to treat subgroup, had about a 20% response rate and about a 20% stable disease rate when MEK inhibition with cobimetinib and anti-PD-L1 antibody with the atezolizumab were combined. Now, you can see that there certainly are toxicities from this approach. Cobimetinib as a MEK inhibitor can cause rashes and diarrhea. And of course, there can be toxicity from checkpoint blockade. But the grade 3 toxicity was low, and this led to a very exciting phase 3 trial, which is currently awaiting reporting. Next slide. And so this has led to a broader thinking about whether we can use checkpoint blockade in combination with other checkpoint molecule blockade or other targeted therapies or chemotherapy in patients with advanced microsatellite stable colon cancer. And hopefully when the slide comes up, you'll see what these studies are. But essentially, the directions are several-fold now. The first question is, even for patients that are MSI high, can we move it up earlier? Because the current FDA approval is for patients who have been previously treated and have had progression of disease on a platinum-containing regimen and an irinotecan-containing regimen. So can we move it up earlier? Could it be first line? Is it as effective as systemic chemotherapy? So that's a Keynote 177 trial. There's also interest, as I said, in combining with chemotherapy in the first line in microsatellite stable or microsatellite unstable cancers. And then finally, even in patients with more refractory disease, is there going to be an impact if we combine checkpoint blockade with chemotherapy and anti-VEGF antibody? There's some preliminary data that suggests if one uses checkpoint blockade with anti-VEGF therapy, there may be enhanced efficacy. So on the next slide is just, again, a little bit more information about some studies ongoing and the approaches that are happening in MSI high and microsatellite stable cancers. And so once that shows, I'm going to also have it stay up there for just a brief second before we move on. Not sure I see it yet here. Okay. The one other study I want to point out here is that there is some interest that if you can't activate a T-cell response, and if right now CAR T-cell therapies are relatively early in colon cancer, that perhaps we can promote T-cell interaction with tumor cells by using BiTE molecules. They bring the T-cell and tumor cell together, and it is hoped that by doing so that the T-cells, if you can apply checkpoint blockade, they'll be ready to kill. So I'll stop there and allow Andi to take the helm from here.
Awesome. Thank you, Mike. This was excellent. So I do think that ending on these last two slides, which really talked about the early studies of interest as well as what is in the field based on the other types of trials, are super exciting. So Mike, one of the things that I want to ask about before I punt into my discussion particularly is really to think a little bit about, you know, you noted some of Dr. Oberman's work coming into GIASCO, and for everyone on the line, there's basically a couple different perspectives that are happening ultimately that I'm not sure why my slides are advancing, so we'll go ahead and just stop right there. I'm not sure what's happening, but Dr. Morse, with Dr. Oberman's work at GIASCO, which is coming forward, and then we'll have the big ASCO conference this summer. Dr. Overman's work you mentioned as something that's really going to be pretty interesting. Can you say a little bit more about what else you expect at GIASCO in terms of some great, hopefully some great foundations or some new science that you're particularly interested in?
Sure, and so far I think we've seen some titles, but of course I don't have the meat of the abstracts yet to know exactly how exciting some of this is going to turn out to be. But I think the directions that are heading and some of the data we'll probably see involve questions like combination checkpoint blockade. Are we going to see greater efficacy if we combine anti-PD-1 or anti-PD-L1 with anti-CTLA-4? There also are some preliminary studies moving through targeting the other checkpoint molecules, LAG-3 and TIM-3. I think we'll see more data on the concept of BiTE molecules and their combinations. And again, some more preliminary data on using targeted therapies and chemotherapy with immunotherapy. I don't really think it's personally that it's going to be unique to one type of targeted therapy or one type of chemotherapy. I think they probably are achieving very similar goals, that is releasing more antigen, altering the immune milieu in a way that's favorable to cytolytic T cells, and also, perhaps by leading to just more cancer cell death, more inflammation in the tumor, that there'll be more epitopes that are T cells can be exposed to, so more T cells that can attack the tumor, and also create a more permissive environment for T cells to truly traffic to the tumor by altering the vascularity, for example, with anti-VEGF therapies, and also promoting the right type of inflammatory environment, the more Th1 type of environment as opposed to the Th2. Also, there's studies ongoing that are trying to look at depleting some of the inhibitory T cells, like the regulatory T cells, and also trying to deplete myeloid-derived suppressor cells, which also tend to create an environment that's not hospitable to cytolytic T cells.
Okay, great. So Mike, what about, and I know you've kind of talked about different contextual environments and inhibitors and the like, what about, as people think contextually also about the microbiome, where are we with that right now, and what do you expect over the next year? What would you like to see happen in this area to have more definitive information about its impact on treatment?
Right. This is one of the really critical areas of immunotherapy right now, because this idea that everything is about the T cell and the tumor, and at least we now understand that other elements of the tumor environment are important, it sort of ignores what else is going on in the body. It's still thinking about things in a small microcosm. But of course, we have a large number of bacteria in our GI tract, particularly in the colon, and we know that these bacteria interact with the host. And there were some interesting observations just even a few years ago. We and others noted in the laboratory that mice that came even from different vendors, that came from different environments, and of course have bacteria in their GI tract, seem to respond very differently to a variety of immunotherapies. And that seemed a little bit puzzling, because the mice are all genetically the same. They should really behave the same, but they of course have different bacteria in their GI tracts depending on where they came from and where they're housed. And so this led to research now in humans, that we know that the microbiome does affect the immune response, and in fact the efficacy, at least from preliminary data, the efficacy of checkpoint blockade. So the race is now on to find out whether there is a particular bacteria or group of bacteria that are important in the response, or whether it's just a certain flavor of the microbiome. It doesn't have to be a particular kind of bacteria, but just bacteria that have certain qualities. And you mentioned that we're part of Dr. Overman's study, and we will have some data on the microbiome and its influence in the outcome of the data that I showed you before. So I think that most studies now, it's very important to not just collect blood and not just collect tumor, but the admittedly not pleasant aspect of collecting microbiome specimens, stool, to also have to be able to analyze in conjunction with all the other immune parameters that we have.
Great. And there's one question real time to you, Mike, I think that I'll bring forth, and then we can get to part of the other questions on the back end of my presentation. But Evan, who's been a great Fight CRC advocate, had noted and wanted to ask, in terms of what the knowledge base is around the Human Genome Project, is there anything specific that we're starting to really look at in terms of genetic solutions to maybe pinpointing treatment for CRC?
Yeah, there are several areas that have really come out of TCGA. The first is that we're now at a point where we can separate colon cancers, not yet clinically validated per se. We're not routinely doing it in the clinic necessarily, but I hope the future will be this, that we can separate colon cancers into different subtypes. They've been called the consensus molecular subtypes, CMS, right now. And there is a subgroup of tumors that are either MSI high or have an MSI high-like genotype. And those are clearly the group that have a very good response to checkpoint blockade. And we're learning more about those. One recent finding that has been the burden of mutations within the tumor. It's often called TMB, the tumor mutational burden. And that those tumors that have higher mutational burdens tend to respond better to immunotherapy. So we're trying to understand, well, what that cutoff should be, and does it depend on the cancer? Some people have suggested the cutoffs are somewhere between 15 and 8 mutations per megabase, for example. And these are data that we can obtain on some of the assays that are submitted to companies that do this type of sequencing. So I think we'll get more clarity around the number of mutations that are important, and also the subgroups of patients who have a molecular makeup that's more likely to respond. I think the other area that we're starting to get, but it's really going to require a much broader exome sequencing, not just for the genes associated with tumors, but really more whole exome sequencing, is what types of immune-related molecules and what polymorphisms in them are associated with better outcome. Again, we want tumors that have a more Th1 type environment, but there are people, of course, who naturally can have that environment more likely than those who don't, based on polymorphisms in their genes. So this is starting to be teased out as well from the TCGA and other attempts to sequence the genome.
Okay, wonderful. And so with that, we're going to take a little bit of a shift and talk a little bit about the real advocacy and nonprofit research domain as it relates to helping supplement this work. And I just want to take a step back and say, I think what Dr. Morse has actually presented is really exciting information, and the body of this growth and work is actually really culminated. Mike, would you say that we've probably seen the biggest explosion in this work in the last 2.5, 3 years?
Oh, I mean, every day it's exploding more and more every day. Even in colon cancer, where again, other than MSI high, things have lagged a little bit. You just see studies that have really piqued people's interest, like the cobimetinib, the atezolizumab, have led to many companies and institutions to really jump in this whole idea of how we're getting closer to being able to target microsatellite colon cancer, and it's accelerating.
Right. So, and I think that that's really a good, primer for what I'm about ready to talk about, is that when I actually started working with Fight CRC in a more deliberate fashion, one of the advocates, Gordon Cole, and his family had left a legacy gift, and had identified that he was on an early immunotherapy trial, but I think he just had a gut feeling of sort, no pun intended, that this was really the direction where science needed to go. And he felt such that he left money to Fight CRC, and with Angie's leadership as our president, and Nancy Roach really thinking about experts in the field to help convene work in this area and to advocate, it really was thinking about how do we look at immunotherapy as standard of care. And I think that's the part of the work that we're really trying to move forward, because we've seen great gains in lung cancer, melanoma. And as Dr. Morse just mentioned, we've seen this explosion every day, but that even has happened in the last couple of years. So when people ask me recently on random, trains, planes, automobiles, on wherever I am in a given day, are we really making progress? And I can say, I feel like we've made more progress in the last 2 years in colon cancer treatment than we have in 10. And so not to say that there weren't great gains in that time, but to say that, I think immunotherapy is rapidly becoming a standard of care as we start to finesse all of these specific types of treatment approaches and matching those to the patients is something that's really happening. And that really happened, I think, with the Fight CRC helping push the envelope with even thinking about the convening of experts. And so what I want to say, of course, is that Young Lee, Mike Morse, and others had really been working on, some of these key ideas. But one of the things that we were able to do at Fight CRC with Cancer Research Institute was also then to have a critical discussion, as at the time we were starting to see some really great advances, pulling together as a neutral convener clinicians, advocates, industry experts, and scientists to ask what are the most salient themes related to immunotherapy, and where should we be going if we're going to do something that would be a bit of a blueprint and a state of the science. And I think Al Benson, who's been one of our medical champions, medical members, Young Lee, Mike Morris, Chris Heary, and others, really got behind this idea and said we need to really have this blueprint, which would be people who may or may not know great information about immunotherapy. What should you be thinking about in research? What should you be thinking about in clinical practice? What should you be thinking about in policy? So we're really excited that we started that effort about at the end of 2015, December, and have actually in that time propelled, where we went ahead and had a working group where we've met over the last 2 years. And in that time, we've developed a framework, which has been a publication that was released a couple months ago through AACR. I'm not sure why my slides want to go ahead and move forward, telling me to move fast, I suppose. Ultimately, that we have a framework where we really have in the literature base right now an idea to help folks really think about how do they really think about immunologic factors to address specific treatment issues, bench science, but also then really thinking from the patient perspective, what are we using in terms of knowing your biomarker and making that specific connection for the right treatment that is right for you. So when we were really working, when we were moving forward with the efforts and where we landed, and I know Dr. Morse had mentioned this, a lot of our ideas were really to think about that T-cell response that you heard about, and then also then, of course, thinking a little bit beyond MSI patients, where we've seen a lot of these real advances. What are we looking at the failure of the checkpoint blockade as it relates to the MSS work, and how are we going to really start to continue to work towards advancing not just microsatellite unstable disease and biomarker of that nature, but the MSS population. And we're spending a lot of time really addressing this as we move forward. So it's really some of the main issues that we're really addressing through these papers, through this work. And it's also really led, again, to thinking a little bit about what are these factors that we need to consider. So what I want to say about all of this is that Mike has done a phenomenal job of talking about the research. I've talked about the state of the science. We've talked about where we're going. But I think one of the biggest issues that have come out of this discussion are also knowing the MSI status of patients. And I think one of the things that we're really sensing is that even through some of the latest polls, research, and data is that we still aren't necessarily finding uniform MSI status of patients in biomarker testing. That's been a huge push from the advocacy organization. Fight CRC and others have really looked at knowing your biomarker, large campaigns, even to try to ensure that people are clear about their MSI status so they understand which mode of therapy might be best, whether it's a complete pure immunotherapy trial, combination trials, but without knowing the status, it's oftentimes really hard to link the patient to the correct linkage. This is one of the biggest policy issues that I think Fight CRC is going to be facing from a medical realm. realm in the next year. And it's something that we're very excited, even coming out of this last week's early onset meeting with National Roundtable and many other partners, to say that this is a policy area that we need to advance. And so much of our work, so much of the patient's information and where they go, and being able to know and to take control of understanding disease and what the implications are, really are completely really harnessed in understanding MSI, and this is something we're going to continue to move ahead. What I'd also like to say is, in addition to creating that blueprint, is that we were also able to we are able to also continue as a convening expert, or our convening group for the experts, I might say. So Fight CRC, as a neutral convener at the outset, was able to cull folks together to develop this blueprint, to really help look at connections amongst different stakeholders. But our real cry from much of the community has been asking that we can continue to convene this sort of work. And so, as Dr. Morse mentioned, at GIASCO in San Francisco, coming just in about a bit of a month, I guess, or so, we will be actually heading there, and we will be actually convening a work group around some of the specifics that we've heard today, and how do we advance this work. And one of those specific areas is really around MSS and working through some of the initiatives that we had. And I think early on in our slide deck, we had discussed, some of the resources that are available to patients. And what I do want to just note, of course, is that Fight CRC has put a good bit of resources in the MSS Trial Finder, working with Tom Mercillier, many of the experts, even Chris Heary, who's helped with that work, inform that work. Maya has been a member of the Fight CRC team and medical science review team in conjunction with Tom. And these are the sort of actual products that we're having coming out of these work groups that also are relevant to patients. So all that to say, is that we're working on really informing a number of the initiatives that Mike talked about, making sure to move the science that's really applicable to bench side. How do we help clinicians and teams understand this to move it forward, to advance research, the policy issues around the MSI, and then thinking of how are we even helping patients linked to clinical trials with things like the MSS Trial Finder, which we've talked about prior, and is definitely a great resource. We're going to continue to advance that work when we convene at GIASCO, and continue to engage with data experts, technology experts like Flatiron, as well as our great scientific team members such as Cancer Research Institute, the American Cancer Society, and the NCCRT partners. So I really wanted to give everyone a bit of a framing that as the science continues, that there's a lot of great levers that can help me to promote general clinical care, policy initiatives, and great resources for patients. And to let you know that this is something that will continue to work, and we're very excited about that. So I know that we have a number of questions, and I want to actually go ahead and jump to one of the things that I want to specifically talk about is that we will, one of the questions that's coming up a good bit right now is, Mike, and I think you and I had talked about this before, if patients at this time, if they're newly diagnosed, this is a two-part question, if folks are newly diagnosed and they're not sure what their MSI status is, what should they do? And if someone has actually completed therapy but understand the importance of MSI therapy, what kind of impact will that have for them long term? And what is your suggestion for people who might be outside of the treatment phase of their care? Mike?
It's considered standard, if you look at guidelines, NCCN guidelines, for example, that everybody with colon cancer should have testing starting with their tumor for the microsatellite stable, for microsatellite instability. In other words, what their MSI status is. Originally, it was, just limited groups of patients. We tried to predict who might be positive. We've really moved beyond that, and we're just testing all tumors now. In somebody who has already been through therapy, but never tested, it's still very appropriate to do it. At a minimum, we're trying to begin the process of understanding whether somebody might have Lynch syndrome. We always start with the tumor, and then if the tumor does have microsatellite instability, the next question is, could it be Lynch syndrome related? And so even if you've been treated, it's very reasonable to have it done just for that reason alone. Certainly, patients who are diagnosed in the past, maybe had been treated, now have unfortunately relapsed, absolutely it would be critical to have it done now if it wasn't done in the past. And certainly all newly diagnosed people, we're certainly trying to have it done.
Okay. So how do you feel, I guess, Mike, when you're thinking about what's currently available and on the market, and you talked about a couple different approaches, I think your last two slides particularly about trials as well as approaches that you think are going to be really the most kind of game-changing pieces. Is there anything that you think is particularly coming up and on the market or going to be coming on the market and available to patients that will really change the course of care? Anything that's going to be timely, or you have a timeline on something that you think is coming?
I think a lot's going to depend on these kind of studies I mentioned, some of which are going to be reporting out in the next year or year and a half that may make a big change. Right now, you know, we of course have had approvals for checkpoint blockade in the MSI high patients. As far as, you know, other drugs in colon cancer in general, as we do more sequencing, we understand their amplifications in HER2, for example, and so you know, we're looking at whether additional targeting can be done. But from the immunotherapy sphere, I say hang on, because I think some of those exciting results will materialize in a year or so, and we'll have new directions.
Okay, so changing this just a smidge, you know, the idea that, you know, knowing tumor markers and the like early on can be helpful, and even in germline, knowing something like a Lynch syndrome could be diagnosed. Is there any thought, Mike, that immunotherapy would actually be used as sort of a prevention as opposed to curative?
Yeah, it's a fascinating thought. If you have a high risk of known high risk of developing colorectal cancer, could you prevent it entirely? I mean, right now the mechanisms for doing that are, of course, identifying people who are at high risk, making sure they're having appropriate screening done. In some instances, this would be more typical for somebody with long-standing ulcerative colitis or known FAP syndrome. Some question about whether people even should have a resection done of their colon. These are all difficult discussions that certainly should be held with hereditary cancer counselors. But I think that it's going to be a ways before we entertain doing this as a preventative. First, we have to look at the data for adjuvant therapy, and these studies are starting to occur now. And I think if there's utility in earlier stage colon cancer and preventing recurrences, I think that brings us one step closer to thinking about prevention. The only concern I have is with the current drugs, they certainly have risks. We didn't spend a lot of time talking about that, but the autoimmune events can be quite serious and in rare instances can be lethal. So I think we have to be very careful when we have people who may have cancers that we're trying to prevent, but we obviously don't want to cause undue harm to people as well. I suspect that if we do get into prevention through the immune system, there'll probably need to be approaches that have a lower risk of toxicity.
Okay, so the other question that's come up several different times, and I know we hit on a couple of those, is why it works in some people and why it doesn't work in other people. So if we really look at that, there's the MSI status, so MSS, MSI, based on those pathways. You mentioned the microbiome. There's other contextual issues. There's ultimately just, the immune status, and to your point, when the drug can be helpful, but when it also can really sort of turn on the body, or the body's defenses really kind of turn on it on its own. In addition to that, are there any other things that you say that, like, when you kind of like look at its simplicity, like in the box of why it works and why it doesn't work, anything else, Mike, that comes up for you?
Well, as you know from all the discussions we had around this framework that you described, it does seem to be very critical about whether there are T cells present in the tumor or not. Tumors that don't have T cells present, it's very unlikely that there's any substrate for the checkpoint blockade to work on. So I think that there's a big gap in our understanding of what happens from the time a T cell is activated till how does it actually get into tumors, and what is limiting T cell infiltration beyond the things we know about? Are there things about the stroma, for example, or the vascularity of the tumor that affect that? And so I think that in addition to studying other checkpoints and ways to interact with them, and studying other ways to activate T cells like cancer vaccines and activating antibodies, I think we definitely need to understand a lot better about T cell trafficking to tumors and getting through the tumor environment.
Great. And so, Dr. Morse, I think we're going to hopefully have a chance and opportunity to connect at GIASCO with the meetings we have coming forward. And of course, we'll continue to convene with you and the roughly 25, 30 other folks about where we're going next. But there has been a question that came in, and I think that this is probably timely. There was a lot of oomph with the Moonshot program and initiatives. The NCI we met with last week, Kathy from NCI, Ned Sharpless, we've gotten a chance to connect with him. And there's a number of folks that talk about the exciting opportunities, and that we have an NCI proposal around genomics, CRC prevention strategies, and even from the Moonshot looking at the psychosocial implication of genetic testing and genomics as it relates to some of this testing. So those were three core areas that they really talked about advancing CRC work. But Mike, what is your feeling that if, as an advocacy organization and with advocates on the line, what do you feel, advocates should really have on their radar as we speak to, policymakers and represent the research community as advocates? What kind of message do you think folks should be sharing?
Well, the first is that everybody probably knows somebody who's had colon cancer. It's a common enough disease, and it really requires a lot of attention. It's tended to lag in interest from funding mechanisms, and in the public's eye, it's tended to take second status to some other malignancies. So obviously it's important to continue to remind people it's a very common cancer. It's becoming a little less common, partly because of screening. But even with intensive screening, there are going to be people that unfortunately develop it and develop advanced disease. I think the second is to continue to emphasize, as we should across the board with all malignancies, with all diseases, is that the science behind many things is still not completely understood, and obviously continued efforts to understand even the basic biology, that not everything has to be about the newest outcome, that thinking very broadly about science and about how the basic mechanisms of whether it's the immune system or other aspects of the development of neoplasia work. And so I guess this is my way of saying, basic science, just like clinical science, counts importantly. And I think the last thing is to remember that, as we said, things are accelerating, and, we're now expecting breakthroughs faster and faster, that it's difficult to have patience if you do have a cancer or have a loved one who has it, but realize that we are getting at a much rapid rate the understanding of the research's understanding of the basic biology and about how to better apply these therapies, and so therefore we really have a lot of information that's moving forward very rapidly, and yet the rate of people entering clinical trials is still very low. And we're never going to be able to answer some of these questions that are coming faster and faster and faster until we can break that bottleneck. I think that needs to be done in two ways. One is people who can participate in studies really should try to seek those studies out. We need to make them more accessible to people at more locations. We need to make the barriers, the inclusion-exclusion criteria, a little bit easier for people to participate. I think on the regulatory side, I think there needs to be, as there already is, a more rapid approach to looking at the data, getting drugs approved more quickly, sometimes on limited data, with the understanding that more data will have to come later, but to not hold anything up if it's very promising. So basically, taking the explosion of data in the early in the preclinical realm and in the early clinical trials, and really powering that through is quicker than we ever have before through those very important clinical trials to get drug approvals.
Great. And I think that, one of the things that we were looking, and one of our medical advisory board members, Dan Dixon, who is with us, had gently reminded us that even over the last 3 years, we'd seen about a 20% decrease in CRC funding, where we've seen other malignancies, as you know, that actually have seen an increase in the aware of 15%. And so I think your point is well taken about how to really move this needle on some of the advancements, because we're making great ground, but we really want to make sure that that pay line of sort to support bench science as well as clinical and translation comes through. So with that, I do know that we are 2 minutes over time, so I'm never quite on time. I want to thank everyone for joining in today. We tried a little bit of a new platform here at the university, but really enjoyed the time. Advance, if there were any technical difficulties, I think we got most everybody on, and super fun. And I want to say again a huge thank you for Andrew, Danielle, Elizabeth from the communications team, do a fabulous job getting the word out. Reese, as well as Dr. Morse, who've really been behind the scenes making sure that we've got materials in play. Kirsten Legrees, Ashley Rankin from here at School of Public Health, helping make sure that we're all moving and grooving. And Michael Sola for always helping be our back support and doing some fantastic tweets. I know I haven't really looked at Twitter for a little bit, but know he's there. So next, I just want to remind all of you that the Research Advocacy Training and Support Program, we will be going through a cohort, a new group of folks that we're including in the RAS program. We're going to continue to do a series of webinars, patient education opportunities, and really just invite you also to even think about and joining in the Trial Finder, finding resources, as well as our grassroots advocacy program. Thank you. Have a great day, and please let us know if there's any specific questions or concerns that you might have. So thank you so much. And that chart is from me.